Good morning, students. I am Dr. Meenal Trivedi, Principal from HVHP Institute of PG Studies and Research, Kadi. And today I am going to explain about the mechanism of transcription in prokaryotic organisms. Students, as you know, transcription simply it is known as a synthesis of RNA using DNA template. Basically, we understand that during the mechanism of transcription, only mRNA synthesis occurs. But it is not like that. Synthesis of any kind of RNA molecule using DNA template, it is known as a transcription. So that in this mechanism, the genetic information is transferred from DNA to RNA. If we compare the transcription mechanism with the replication mechanism, here you can see the similarities between the mechanism of replication and transcription, as both the processes need DNA as a template. Second, there is a formation of phosphodiester bonds in both the cases. And third, in both the cases, the process occurs into the 5' to 3' direction. Whereas, if we see about the differences between the mechanism of replication and transcription, you can see that in mechanism of replication, the parental DNA gets separated and both the strands of DNA act as a template. Whereas, in case of transcription, as the RNA is single-stranded, only one strand of DNA acts as a template during the mechanism of transcription. The substrate used in replication are deoxyribonucleoside triphosphates, whereas in RNA synthesis, ribonucleoside triphosphates are used as a substrate. There is no need of primer in case of transcription, whereas it was needed during the process of replication. In mechanism of replication, we have seen that dozens of various enzymes were used and RNA polymerases were the key enzyme which were synthesizing the DNA molecule. Whereas the entire mechanism of transcription is carried out by a single multimeric enzyme which is called RNA polymerase. After the mechanism of transcription gets completed, there is a synthesis of RNA molecule, whereas at the end of DNA replication mechanism, double-stranded DNA, the entire genome gets duplicated. So this is basic difference between the mechanism of replication and transcription. Now, before we study the mechanism of transcription, we should understand what are the enzymes needed for the mechanism of transcription, what DNA act as a template, and about the transcription unit. So first we are starting about the DNA template. As we have discussed that during the replication mechanism, the entire genome gets duplicated. But here, in case of transcription, only a small portion of the genome, which is basically known as a gene, only a gene is get transcribed wherever a particular gene product is required by the cell. And these structural regions which are transcribed into the RNA, they are known as a structural genes. Now, this gene is double-stranded. Among both the strands of DNA molecule, only one strand acts as a template, whereas the opposite strand of the DNA does not act as a template. The template strand, which is used as from which the RNA synthesis takes place, it is known as a non-coding strand. It is also known as a antisense strand. Whereas the strand which is complementary to the template strand, that is known as a coding strand, and this is the strand whose base sequences specify a particular amino acid sequence of the encoded protein. So that this strand is also known as a sense strands as well as coding strand. You can see into this picture, this is the presence of a template strand, and complementary to the template, there is synthesis of RNA molecule. You can see over here that in your double-stranded DNA molecule, the opposite strand of your template, that is this 5' to 3', and you can see the RNA sequence which is having the similar sequence of this strand. So here this is a coding strand which is non-template, this is your template strand which is non-coding strand, and complementary to the template there is synthesis of RNA molecule takes place. Now, Transcription is entirely asymmetric mechanism, as only one strand of DNA used as a template in the transcription, but the coding strand does not act as a template in the mechanism of transcription. In this mechanism, both the strands can be used as a template. So transcription direction on the different strands, it is opposite, and this particular feature, it is known as a asymmetric transcription. You can see into the next picture, you can see two regions. Here, concentrate over here, the lower strand is a template, the upper strand is a non-template. As we have studied that the non-template is known as a coding strand, the lower one which is a template, and complementary to that RNA synthesis takes place. Whereas into the same gene, into the other region, there is possibility that the template is at upside, coding strand is a downside. So you can see over here, this is entirely referred as a asymmetric transcription mechanism. After that, the second important component we need to study before we understand the mechanism of transcription, that is the RNA polymerases. The enzyme which is responsible for the synthesis of RNA molecule. This is also known as a DNA dependent RNA polymerase, because from the strand of DNA, these enzymes synthesize the RNA molecule. The prokaryotic DNA polymerase is a consisting of multiple subunits. So this enzyme, it is known as a multimeric enzyme, having the approximate weight of 480 kilodalton. And this single bacterial RNA polymerase synthesizes all types of RNA molecules in bacteria. Whereas, if we concern about the eukaryotic system, eukaryotes have the three kinds of RNA polymerases, and each is responsible for the synthesis of different RNA molecules. If we see the structure of bacterial it is a bacterial RNA polymerase holoenzyme. The enzyme consisting of 5 different subunits, 2 subunits of alpha, 1 subunit of beta, and 1 subunit of beta prime, that synthesize the core enzyme. And to this core enzyme, sigma factor associate, and this convert into the holoenzyme. So you can see over here that this is the bacterial RNA polymerase consisting of the core enzyme consisting of 2 subunits of alpha, one is of beta, one is of beta prime. That is known as a core enzyme. This core enzyme binds with the sigma factor. Resulting component, it is known as a bacterial RNA polymerase holoenzyme. Now every subunit has different molecular weight and having a different function. As the core enzyme consisting of 2 subunits of alpha, this alpha subunit each consisting of 36.5 kilodalton molecular weight, and it is determined to determine the DNA to be transcribed. The beta subunit having a molecular weight of 151 kilodalton, and that categorizes the polymerization of RNA molecule. The beta prime subunit, it is having the activity just like the helicase, which binds and open the DNA molecule by break down the hydrogen bonds. The sigma subunit, when to the core subunit the sigma factor associate and resulting component, as we have discussed, it is known as a holoenzyme. And this sigma subunit has a function to recognize the promoter, attach to the promoter, and to initiate the RNA synthesis. So sigma factor play a significant role into the initiation of the transcription process. Prokaryotes use the same RNA polymerase to synthesize all types of RNA molecule. Now, second important thing we need to understand before we study the mechanism of transcription, it is known as a transcription unit. As we have discussed earlier that during the mechanism of transcription, not the entire genome is involved. Only a specific gene is transcribed, that we called as a structural genes. So basically, in short, we can say the transcription process must start at a specific point and it must get terminate after at a specific region. So this transcription unit consisting of nucleotide sequences into the DNA which encode a single RNA molecule along with the other necessary sequences required for the process of transcription. Normally, this transcription unit consisting of major three components: a promoter region, an RNA coding sequences, and a terminator region. Promoter play a significant role to initiate the process of transcription. The RNA coding region is the actual sequence which will be transcribed to synthesize the RNA molecule, and the last one is the terminator sequence that play a significant role into the termination of the process of transcription. So you can see that there is a presence of promoter. At the center there is a presence of a RNA coding region, and it is followed by the terminator. If we focus about the RNA coding region, the promoter is present ahead to the RNA coding region. In molecular biology, it is known as the upstream towards the RNA coding region, and the termination sequences is present downstream to the RNA coding region. The process of transcription starts at the promoter and move towards the terminator sequences. The actual movement, it is known as a movement of transcription bubble. So you can say like that starting from the promoter region, the transcription bubble moving downstream towards the gene at the terminator sequences. So this is the way how the process will get continued. Each transcription region is called operon. So you can see over here the promoter followed by RNA coding region, which is actually a gene that will be transcribed, and the terminator sequences. And these entire component, it is known as a promoter. Each promoter consisting of structural genes and regulatory sequences for the mechanism of transcription. Now we have discussed three components of transcription units: promoter, RNA coding sequence, and the terminator. So first of all, we understand about the promoter. It is important and everyone should understand what by definition promoter is known as. So a promoter is a DNA sequence located immediately upstream towards the RNA coding region, and this is the sequence to which transcription machinery bind and initiate the process of transcription. So this is the promoter at which the RNA polymerase holoenzyme binds. It is a very important component to control the mechanism of transcription. Although promoter vary from different genes from in prokaryotes and in eukaryotes, every promoter consisting of certain common sequences, and these are certain similar sequences which are recognized by the RNA polymerase holoenzyme, so that RNA polymerase holoenzyme can come to know that it need to bind over here and initiate the process of transcription. So this bacterial promoter, they consisting of two sequences at -10 region and -35 region. These are the two important consensus sequences present into the bacterial promoter. You can see over here, this is the structural gene, and upstream to the structural gene there is a promoter region to which RNA polymerase bind and initiate the process of transcription. Now, if we consider this one as a starting point of your RNA coding region, if you are moving to the upstream location, you can count the nucleotide as a minus scale, -1, -2, -3, likewise up to the -50. It has been mentioned over here. So at -10 region, there is presence of a specific sequence, that is having a similar sequence into almost all bacterial promoter, and this kind of similar sequences, they are known as a consensus sequences. This -10 consensus sequence, it is known as a Tata box or Pribno box into the bacterial promoter. The second consensus sequence is found into the -35 region. The sequence is TTGACA. So these two important consensus sequences give the signal to the RNA polymerase holoenzyme to attach at this location. If any mutation occurs into the consensus sequence, RNA polymerase cannot recognize this promoter, cannot bind to the promoter, and no transcription gets initiated. So these are the two important consensus sequences present into the prokaryotic promoter. The -10 consensus sequence, it is known as a TATAAT, -1035 sequence, it is known as a TTGACA. And this -1030, this consensus sequences, they are recognized by the promoter, recognized by the RNA polymerase and bounded by the sigma factor of RNA polymerase holoenzyme. Now, you might be thinking, what do you mean by consensus sequence? Over here, various promoter sequences has been given. You can see over here that in this -10 region, in the first one the sequence is TATAAT. In the second one again it is TATAAT. In the third one you can see that it is TATAACT, TATGTT, TATAAT, TACTGA. Means there is certain sequence similarity found over here, and these are known as consensus sequences. This is the way how the transcription unit is working. And next process is the mechanism of transcription, divided into three phases: initiation, elongation and termination. Students, we will discuss the mechanism of transcription in our next lecture. Till then, take care of yourself. See you.